Hello everyone, welcome to another lesson. I'm Anna Rajan. Today I'm going to start a new series on genetics and audiology. Here in genetics, I'll be explaining the general terms and few terminologies and nomenclatures that you need to understand before going into different types of hearing losses that are genetically occurring or hereditary. So, genetics plays a major role, and understanding genetics will help us understand auditory system better. If you look into the epidemiology, out of 1,000 births that are happening, in the 2 to 3 children are born with a permanent hearing loss, and the interesting part is that 90% of the parents are normal hearing parents. So let's look into the etiology of hearing loss. Hearing loss can be caused by 50% of genetic reasons and the percentage of environmental reasons. So when it comes to genetically caused hearing loss, in that 70% of cases, 70% of times, it is syndromic. It is associated with a syndrome. So a child might be having a syndrome and one of the feature will be hearing loss. And secondly, it can be non-syndromic. 30% of the genetic hearing loss cases can be non-syndromic. In the syndromic cases itself, there are different types of hearing losses which can be occurred autosomal recessive, or it can be autosomal dominant, or it can be either sex linked or mitochondrial. We'll be talking about what these terms are, what is autosomal, what is recessive, what is X-linked, what is mitochondrial mutations. All this will come in the next few slides. So, when it comes to the basics, the genetic information in human is carried in a DNA. So, DNA is a deoxyribonucleic acid. So nucleus of this DNAs or nucleus of any cell organelles or a mitochondria of any cell organelles have certain structures called nucleotides. So these are the basic structures, nucleotides. Each nucleotides, if you see, it is made up of certain materials. So each nucleotide is made up of a sugar component, a phosphate, and 4 chemical bases. So the backbone, the outer structure of any, let it be a DNA or a nucleus or a mitochondria, it is formed with the outer structure is formed by sugar and phosphate, and the inner connections are the chemical bases. So there are basically 4 chemical bases that we can see in a nucleotide. Those are adenine, thymine, guanine, and cytosine. So basically, these nucleotides pair up to form the double helical structure. So we always say that DNA has a double helical structure which looks like this. So it is formed by a pair of chemical bases. So it has a pattern. Adenine always pairs with thymine or vice versa, and guanine always pairs with cytosine. So these combine to form the double helical structure. That means whatever is there in the one side, it has a complementary side. So whenever there is, for example, when there is a thymine, there is an adenine coming in the other side. And when there is a guanine, there is a cytosine, or uh, like vice versa too. So in a DNA, there is approximately 23,000 genes. So it has so much of information. Each genes can form each proteins. Okay. So when it comes to genes, um, like if you take a DNA, each part, if you take each segment, if you take based on the functions, we can categorize that segments into genes. Just like how I told, we have approximately 23,000 genes. So each genes have amino acids. These amino acids are arranged in certain pattern to form the proteins. Amino acids are arranged in certain pattern to form proteins, and this process is called as translation. The process of arranging the amino acids into a pattern to form the proteins. So these proteins are basically protein shows the functions of each genes. So there are certain what to say, the segments called exons. Exons are the ones or the codes. It's a coding region. If you take a DNA, it has certain parts which can be labeled as exons. What this exons does, or these parts of genes does, is these exons instructs the amino acids how to arrange or how to form the sequence. So basically, it instructs the amino acid to how to arrange, how to organize itself. And there are codons. What does codons do is, like, codons produce specific amino acids. For example, there are start codons. So at the beginning of a gene, if you have a start codon, or it starts coding a protein. And when it comes to a stop codon, the stop codon stop the process of coding the protein. So we have start codon, stop codon, codons, and there is something called as introns. So if you see here, you can see certain gap between both exons. Those are called introns. Introns are nothing but these are the area which are not coded. Nothing is happening here. So here certain proteins are getting coded. That means certain amino acids are instructed to form in different shapes to form a structure to make a protein. And it begins with a start codon, it ends with another codon. That means it starts the coding procedure. And that is how it produces the amino acid. And in between, there are certain areas which is not getting coded, which has nothing in it, no information. So those are introns. Okay. And we have something called as chromosomes. So if you see in a DNA, we have, or if you take a human genome, in the DNA, we have 3 billion base pairs, like how we saw AT, CG, just like that, we have 3 billion base pairs. So all of this information is organized into 46 chromosomes. So the organization of these base pairs is called as a chromosome. So in humans, we have 46 chromosomes, and these are arranged into 23 pairs. So we basically have 23 pair of chromosome. And in that 23 pair, 22 pairs of chromosomes are called as autosomes. This is same in, like, both the genders, males and females. It's same in everybody. This 22 pair of chromosomes is similar. But there is one pair of chromosomes which is different than male and female. Those are the sex chromosomes. So these chromosomes basically define what you are, like whether you're male or a female. So out of the 46 chromosomes, that is the 23 pairs, 22 pairs are same, and these are called autosomes, like I said. So we have autosomes and we have sex chromosomes. So if you take a person, for example, if you take me, I have 22 pair of autosomal chromosomes, autosomes, and I have one pair of sex chromosomes. Since I'm a female, my sex chromosome will be XX, which you have already studied in your biology lessons as well. And for a male, the sex chromosomes are in a pattern XY. Okay. And based on where the chromosome is placed, or how the chromosome, the type and structure, and based on how you define your chromosome, that is what we call as karyotype. So the structure, the position, everything about the chromosome is called as a karyotype. And based on where it is positioned, we can say this is the chromosome. We can give a number. So loci is basically the position of the chromosome, can be defined by a specific numbering of the chromosomes. So if you see a chromosome has a short arm which can be named as P, and a long arm which can be named as a Q. So this is P and this is Q. So it is basically like two different parts, a short part and a long part. Short arm is called this, it's we term it as P, and Q is for the long arm. And these two P and Q area, or the short and long arms of a chromosomes, are connected by using a centromere. Okay, and the ends of this chromosome is called as telomere. This is the just basic structure of a chromosome. For example, I have given a example here, 13q12.11. So this shows a level of a chromosomes. For example, if in this 13 is the number of chromosomes, so in this 23 pair we can, or 22 pair, we can pair it as we can number it as 1 to 2, 1 to 22. So in that 13th pair, we have the Q. Q means the long arm, and the position is 12, 1, 2. So this is basically um, when you stain and keep the chromosome, and when you stain the chromosome and put it in a microscope, you can see different kinds of bands. So this is band 1 and band 2, and this is subband 11 or 111. So you can call this as 13q12.11. This is how you label a chromosome. And there is something called this genotype and phenotype. Genotype is basically nothing, um, like how I said, 13q or something like that. So, for example, if a DNA is coded in a certain way, how that code is called as the genotype. So, I just told 13q um 12.11 or something. This is a code. This is a code for a gene, or that is just a position of a chromosome. But if you take a gene, a particular segment, it is coded in a particular way. It has a shape. So if you draw a line, if you draw a circle, that has a shape. So that path is called this genotype. But when there is a variation in the genotype, how it is getting represented is the phenotype. So for example, let's make it simple. So a chromosome is basically an information. It's like you're downloading the information into another device or something like that. Just like that, you're carrying the information from your parent into yourself using the chromosomes. Chromosomes is basically has all the genetic information. So this information, if it is, it's 90 percentage of the time it's the same in all of us, or 99.5 percentage, I guess. It's almost similar in all the individuals. That little bit of variation is what makes each one of us different from each other. So for example, a particular code, a particular gene has an information about the eye color. And for example, you're a person with black eye, and I have brown eyes. So maybe in my code it might be different. For example, I'm just taking an example of ABCD is the code for eye color. You have ABCD, I have ABDC. That is the difference that is making. So that is a genotype. So your genotype is ABCD, my genotype is ABDC. So what is a phenotype? Just so since you have a genotype of ABCD, your eye color is black. So that is the phenotype of this particular genotype. That is the ABCD is the genotype. Black color, black eye color is the phenotype, how it is getting physically expressed. So for me, it is ABDC is my genotype, and I am having a phenotype of brown. It is shown as a brown eye color. So all these variations are the ones, or these genotypes and phenotypes. Genotypes are the one which is shown as phenotypes, and these variations are the one which makes each one of us different. So this is not a mutation. This is not an issue. So if it was same, everybody would have looked like a robot, or a product made from a factory. But we all are different because of different genotypes. These are not what to say. It's not an issue. It's these are variations. So when it becomes an issue, it becomes a mutation or a problem. Okay. So, and there are different types of genes. There are heterozygous ones and homozygous ones. So if you see here, it is similar. Both the arms of the chromosomes are same. It has B and B, capital B and capital B. So that means same types of genes comes, then it is homozygous. But here, if you see, it is two different types. Then it is heterozygous, as simple as that. Let's talk about genetic mutation. Like I said, we have different genotypes, we have different phenotypes. So when it is getting copied from a parent to a child or an offspring, or when there is so it is ideally supposed to come like ABCD, but instead it comes as something else. then that is a mutation, and it manifests as a problem, then it is a mutation. Okay. So there are different types of mutations that are seen as listed. So substitution is there. It's self-explanatory from the term. So that means it's substituted by term. I'll come to it. Substitution, deletion, insertion, missense mutation, nonsense mutation, frame shift mutation, expansion. So these are different types of genetic mutations. So for example, I'm taking a sentence here. Okay. I let's have some, instead of a gene, I'm gonna put an English sentence for you to understand. So I have a sentence here which says I have one dog. Suppose this is our genetic code or genotype. I have one dog. So when it comes to substitution, just like how it says, what is gonna happen? Any one of the components is gonna get substituted with something else. So for example, I have one dog. When it gets transferred to me from my parent, it became I have one cat. So one component is substituted with something else. Okay. So this might manifest as a different feature. And next one is deletion. What happens in deletion? A part of our gene gets deleted. So here I have dog, but I don't have that part which says one dog. So that means that one got deleted when it got transcribed. So next is insertion. So insertion means something else is getting added into the code. So I have one big dog. So big is an addition. Just like that, in this, these are an example for you to understand. So just like that, in a genetic code, you get a component added. Then there is something called this missense mutation. Okay, so wherever I have spoken, for example, substitution, I have one cat, I said. So if you see these two sentences, I have one dog and I have one cat, the order is same, the arrangement is same, the number of alleles or components are also same. For example, 1, 2, 3, 4 words. If you take that sentence, here also 4 words, only that one factor got substituted. So such kind of mutation where there is no major change in the structure of the gene or number of components in the gene or alleles in the gene, that kind of mutation is usually known as point mutation. So when point mutation happens, what occurs is like sometimes, I'll show you an example for missense. There is, the sentence is similar, but just and substitution for one has happened. But the in turn, nothing is happening in the phenotype or no major changes happening in the gene. Such mutations are called as missense mutation. And then we have nonsense mutation. Nonsense mutation is when I've spoken about stop codon, start codon and all. A stop codon is a part of the gene, right? It gets added in a wrong position. So suppose I have one dog is the code getting typed in, and in the sentence, if you see the stop codon should come after dog, that means it should say that stop making the sentence. Stop codon puts an end to it. So that, like, just like that, where the amino acids are formed or the protein is formed, where the stop codon occurs, that process of making the protein stops. So here, instead of putting after the dog, what happens? The stop codon came in between after half. So what happened? It just coded half of the information. I have, and it stopped. So this doesn't make any sense. Does that sentence make a sense by itself? No. So that's a nonsense mutation. Similar way in a gene coding happening when amino acids are getting arranged because of the exons, a stop codon suddenly comes and stop that procedure. That protein formation is not complete. So that means the protein is not formed properly. That's a nonsense mutation. It doesn't function properly. Then there is called something called as frame shift mutation. In frame shift mutation, what happens? A segment gets deleted and it gets reversed and gets attached in different region, like an inversion or something. But what in frame shift particularly, what happens is like, if, for example, like we said, if just 3 units of a gene is getting deleted and getting substituted, or some changes is happening as a multiple of 3, because it comes sets of 3, nothing much happens, which the shape, that like the meaning doesn't change much. But when it is not a multiple of 3, what happens is, like, for example, um, we say adenine, thymine, or cytosine, guanine. It's like that. So three segments are going, and just a deletion or a substitution or something happens just for 3 units. It will just happen for that particular segment. But if it is more than 3 or not a multiple of 3, what happens is, like, the entire code below that gets changed. So that is called as frame shift mutation. And there is something called expansion. Expansion is like a specific part is getting repeated itself. So here I have one dog. So the sentence is getting expanded. Just like that, the code is getting expanded because one component or one part or one segment is getting added multiple times. Then there is something called this Mendelian inheritance. I'm sure like all of you would have learned this in your science classes in school. So Gregor Mendel had done a lot of studies related to genetics. So he has given certain patterns for inheritance. For single gene mutations, like when one gene is affected, there can be any of these 3 patterns. It can either be autosomal dominant, autosomal recessive, or a sex-linked mutation when a mutation is happening. So these are, we'll come to this, but you have to see that when it says autosomal, be it dominant or recessive, it is affecting the autosomal chromosomes, which we have talked before. And when it says sex-linked, it is affecting the sex-linked chromosomes. Okay. And before knowing that, we need to know something about pedigree chart. Pedigree chart is something given by Gregor Mendel again. This is a representation of the family history. So, or a pictorial representation of the family history. So, when you look at a pedigree chart, you can understand that how a certain condition or a certain genetic mutation is getting carried over the generation. So, in a pedigree chart, when you see a pedigree chart, the circles represent the females and the squares represent the males. And if it is shaded, it means that they are affected, and unaffected individuals are usually not shaded. And whatever is coming in the same row is one generation. And like each row represents each generation. It is like that. And if you see an arrow beneath any of these affected individuals, that means this is the first person to seek a medical attention for that particular condition. Okay, that's how it goes. So here, just to say, a father and a mother, mother and a father is affected, and they have had 3 kids, 2 boys and a girl, and both the boys are affected, and one got married to a female, unaffected female, and they had 2 boys and 1 girl, among which 2 boys are affected, and this one of this affected individual had gone to get medical attention. This is how the pedigree chart is going. So, like how he has classified, first let's talk about autosomal dominant inheritance. So when you say dominant, it means that something is on power. It's like that. So we have told that we always have chromosomes in pairs. Like we have um, chromosomes, yeah, paired. So for example, a person is having a a pair of chromosome, for example, A and B, and another person is having C and D. In that, A has dominant characteristics, and in the female, D has dominant characteristics. If that A is affected, both the in the pair, both the chromosomes need not have to be same affected to cause a disease. So wherever that affected or the dominant chromosome is coming, whatever characteristics it has, it will show. So that's the basic ideology. So if you see here, I'll explain in a better way. We have a father who is affected here, and we have a mother who is not affected. And if the father is affected, that means his one of the chromosomes, he has 2 pairs, right? For example, one of this, the green one, is affected. That is the dominant chromosome of dominant pair of chromosome or dominant part of his chromosome is affected, then how is the pairing gonna occur? So one pair from one chromosome from here, one chromosome from the mother. So it has an affected pair and an unaffected pair. Since it is dominant, since this is dominant, that child born to that if that is the pattern that is getting copied, he'll be affected. And suppose the second pair of chromosome from the father and another pair of chromosome from the mother comes, the child is unaffected because it is not affected. It is a recessive one. And first pair of chromosome from the father or the affected pair part from the father and one from the mother, again the child will be affected. It just needs one affected chromosome to show the effect. That is the dominant one. Understood? So, for example, here the mother is affected. Mother has one chromosome which is affected. Whichever combinations has that one, it can manifest itself. It may not have to be in pair. That is the dominant. So whenever that dominant chromosome is affected, we call it autosomal dominant inheritance. Similar way, when it is recessive, even though one pair is affected, like you see here, the child can be a carrier, but the child may not be affected or child might not be showing an effect. That phenotype might not manifest. The child might carry the chromosome, but child gets affected only when both the pairs of chromosomes comes together. So in the previous case, chances of getting an affected child is 50% because out of the 4 pairs, 2 pairs might have this chromosome, will have this chromosome, and in that case, it can manifest. So if the child is born with any of those pairs, then the mutation can manifest. But here in this case, the chances are only 25%. So if you see, the chances of child getting the affected plus affected part is only 21 out of 4. So that means 25%. Then there is something called as de novo mutation. This is for you all to find out. Just read upon that term, and if any one of you know the answers, please put it in the comment. Just read upon what is de novo mutation. Okay. And then there is sex-linked chromosome mutation. So this is basically just like how we told, we have 22 pairs plus a sex chromosome. And that if this mutation is happening in that particular sex chromosome, that is when we call it as a sex-linked mutation. So sex-linked mutations usually happens in the X chromosomes, not the Y. I'm not saying it's just from the mother, but if it is coming from the father also, X chromosomes are affected because Y chromosomes carry very less genes or genetic information compared to all the other genes. So very less proteins. So if since it has very less genes, it doesn't affect much. So sex-linked chromosomal abnormalities are usually associated with X chromosomes, since most of the daughters would be the ones who are affected in this case. I think more females are affected. Then there are something which Mendel didn't explain. So those are called this non-Mendelian inheritance. Inheritance can happen in a different way. So one such case is mitochondrial inheritance. Mitochondrial inheritance is, so far we were talking about genes coming from the DNA, but here this inheritance happens through the mitochondrial cells. And since it happens to the mitochondrial cells, females are mostly affected, not females, the affected mothers can give out that mutations, not from the parents. But all other mutations that we discussed previously can come from both the parents, both the father and the mother. But here, mitochondrial mutation usually happens from, if the mother has a mitochondrial mutation, the chances of the child getting the mutation is more. Why? Because during fertilization, sperm loses all the mitochondria. So the mitochondria is getting transferred from the mother. Since it is coming from the mother, so only when a mother is having a mitochondrial mutation, that is getting transferred to the child. Then there is called this polygenic mutation. Polygenic mutation is nothing but it's a combination of different mutations. So for example, like I said, I have a problem with something, but not, not not like that. How do I explain it? Okay, polygenic is like even though one gene is mutated, it will not show us a problem. But when it combines with some other mutations, these mutations together becomes a problem. For example, like I said, eye color is different. For example, my eye color is brown. That is one of that's not a mutation. How do I take it? Okay, just for example, I have brown eye color, and some other chromosome says blind eyesight. But this blind eyesight occurs only when this brown color and this two occurs in one person. Brown and this eye issue occurs in one person, it manifests as a problem. If I have brown color, I won't have eye issue. If I have these particular gene and no brown color, I'll not have eye issue. But if these two comes together, I'll have an eye issue, just like that. This is just an example, okay? So more than one mutation is required. The combination of more than one mutation happens, then only that gets manifested, and it is a polygenic mutation. There are some monogenic mutations are there. These are polygenic one. Then there are called modifier genes. Modifier genes are the ones genes might be in that person, but these genes define how a problem develops, or when to start, how it progress, and how much of a severity it goes. For example, if you take hearing loss, modifier genes decides like what can be the onset of this person might have this modifier gene inside. It might say that it starts with 3 years of age, so the child might develop hearing loss at 3 years of age, and it starts slowly and progress from mild to severe, or it can abruptly start as a severe hearing loss or profound hearing loss. So it all depends on the modifier genes that they have. And then we have multifactorial um, inheritance. Multifactorial inheritance is like there will be two components, a genetic factor as well as an environmental factor. So it's not just because you have a particular type of gene, you might not have an issue, but when it comes along with an environment, it can have an issue. For example, a person is having a particular gene, um, which can lead to ototoxicity, but or when it is combined with aminoglycosides forms ototoxicity. uh, so, for example, there is a person who has this genes and I am there. Even if me and that person takes the same medication, aminoglycosides, I'll not have an issue. I might not get ototoxicity. But if that person takes aminoglycosides, he will get ototoxicity. Why? Because he has a particular gene um, in him. But when it gets combined with the environment, it can form a mutation. So such mutations are called multifactorial mutations. Then there are certain chromosomal abnormalities. These whatever we've solved, all this about the genetic mutations, how it is happening, and then there can be certain chromosomal abnormalities. So such examples are so usually there are certain terms that you have to be familiar with. There is euploidy. We say euploidy. Euploidy is nothing much. Uh, we have told that we have a XX chromosome or we have an XY chromosome. So these chromosomes are occurring in a pair of two. So that pattern is called as euploidy. So in sometimes when it gets coded, it changes in number. For example, trisomy. So if you see here, I'm sorry, it's wrongly marked. If you see here, the red one, instead of two, the chromosomes are made of three components, right? So that is called as the trisomy. So when the trisomy of 21st chromosome occurs, that is when people have Down syndrome. So that is getting manifested as Down syndrome. Down syndrome is basically the trisomy of 21st chromosome. So trisomy is an abnormality. It is supposed to be 2, but instead it is coming in 3. And then similarly, there is something called as monosomy. So instead of 2, it becomes 1. So this usually happens in the X chromosomes in females, and this is usually manifested as Turner's syndrome. And then there is polyploidy. Polyploidy is like instead of 2, it will have multiples of 3 or something like that. For example, instead of XX plus or 4, um, we usually have 22 pairs, like 44 plus XX or 44 plus XY. Instead of that, we might have 69 plus XY or 69 plus XX. That means there is multiplication or like numerous like number instead of two pairs of 2. So usually when polyploidy happens, so humans don't survive. That is when there are miscarriages and all usually happening. Uh, it is lethal to humans. Okay, and there are certain diseases which comes as non-syndromic hearing losses and syndromic hearing losses based on what is happening in it. So we'll be talking about each of this in the upcoming chapter. So um, I know there are different terms. These are just for you to get familiarized with all these terms so that when we are talking about the non-syndromic hearing losses or syndromic hearing losses, it will be easy for you to understand how it is getting manifested. So in the upcoming video, I'll be talking about non-syndromic hearing loss. And in the following videos, I'll try to put syndromic hearing losses or I'll make videos for each syndromes for you to understand easily. Um, so that's all about an introduction to the genetics series. I hope it was clear. And if you have any queries, you can put it on the comments. And you can always put suggestions, and you can always put like suggestions on what topics you want a class on. I'm always ready to do that. And if there are any queries, you can reach out to me. That's all for today. Thank you. Here you have my email in case you want to reach out to me. Thank you.